Hi, and welcome to our lecture on the autonomic nervous system. In this chapter, we're going to take a look at this involuntary motor system and its two divisions, the parasympathetic division and sympathetic division, although we'll actually see kind of something extra at the end. I want to begin here with what ANS stands for. It does stand for the autonomic nervous system, and we want to kind of take a closer look at autonomic to make sure that we remember this correctly. Autonomic comes from the root words autos and nomos. Autos means auto or self. Nomos means law. Therefore, we're talking about autonomy, the self-governance of our body through this purely motor component of our nervous system. So the ANS is going to be providing motor signals only, and these are going to be to those involuntary effectors: cardiac muscle, smooth muscle, and glands. We want to contrast signal transmission from the central nervous system to the effector, comparing somatic and autonomic divisions. Let's go ahead and begin with the somatic division on the right. We know that we're going to initiate this command from a cell body which is located in the anterior horn of the gray matter of the spinal cord. We're going to work our way out through the anterior root, remembering that the anterior root carries all of the motor commands and the posterior root the sensory information. And we travel on this same neuron all the way out to the skeletal muscle, which is our voluntary effector. In the case of the autonomic division, over here on the left-hand side, we again begin in the gray matter. In this case, this would be in a lateral horn, although there are some complications to this we'll cover in just a moment. We are going to go out through this anterior or ventral root, hop onto the spinal nerve, and we are going to exit the spinal nerve, in this case, on a communicating ramus. This shows one particular possibility, which is where we're going to go to this sympathetic trunk ganglion and synapse, that is to kind of go across this neurochemical gap between two neurons and get onto our second neuron here. And we're going to take this second neuron out to our involuntary effector, in this case the smooth muscle of the trachea and bronchi. So what we're going to do, what is different about these two divisions, is that we're going to take a single neuron from the central nervous system to the effector in the case of the somatic division, and we're always going to hop onto a second neuron in the case of the autonomic division. How exactly we travel on those two neurons in the ANS is going to vary depending on the particular application. So we want to get some names so we can talk about these neurons. The first neuron in this ANS pathway is called the preganglionic neuron, and that name should be pretty obvious. We're going to start here in the gray matter of the spinal cord. We're going to travel to a ganglion. So that neuron is mostly prior to that ganglion. Obviously, it has to enter it, and it is going to be myelinated. That actually brings up an important point about these communicating rami. Since this is hopping off on this one over here, this ramus is going to carry myelinated axons, and we're going to call that the white ramus communicans. So we're going to potentially synapse in there onto our second neuron. Our second neuron is called the ganglionic neuron because it begins in a ganglion rather than in the gray matter of the central nervous system. We actually refer to its axon as being postganglionic because the axon will extend away from the ganglion and go towards the effector. This second neuron is going to be unmyelinated, and that will come up a little bit later on. In addition to being able to go directly to an effector, it might hop back on a spinal nerve. If it does, it takes this other communicating ramus here, which would be a gray communicating ramus because it doesn't have myelinated axons running through it. The axons running through it are unmyelinated. So let's kind of get an idea of how this kind of stuff works out a little bit. There is a little bit weird going on in the autonomic nervous system. Our basic idea, of course, always is we have this preganglionic neuron. It is located in a lateral horn. It extends out through the anterior root, gets to a ganglion at some point, and we synapse, getting onto the ganglionic neuron, going out its postganglionic axon to the effector. What you can see here is how this is actually going to synapse once it gets to the effector is interesting. There are numerous of these postganglionic varicosities on this postganglionic axon, and each of them is capable of releasing from these synaptic vesicles neurotransmitters into a gap between this varicosity and where we have our receptors on our effector organ. So in this case, we can see that this is muscle. We have the sarcolemma as the plasma membrane on the outside containing those neurotransmitter receptors. This basically means that we can actually synapse with kind of a long organ like an intestine here, and it kind of makes sense that it's a little bit different than when we synapse maybe with individual muscle fibers. So let's take a look at the two divisions of the ANS. This is kind of the main component of this chapter, is understanding the differences between the sympathetic and parasympathetic divisions. Now, one of the kind of obvious differences we can see here relates to how they are set up. The parasympathetic division has a long preganglionic axon. You can see it myelinated here with Schwann cells that they've placed on this. We're going to synapse within a ganglion. This ganglion will always be near the target organ, meaning we have a short postganglionic axon. We're going to use acetylcholine as a neurotransmitter both in the ganglion and at the site of the effector. In the case of the sympathetic division, we tend to have short branched preganglionic axons, meaning this can actually synapse with multiple things. We are going to then go to a ganglion. This should be relatively close to the spinal cord. Synapse, we have a long postganglionic axon. We're going to release acetylcholine again in the ganglion, but norepinephrine will be the neurotransmitter used at the effector. So let's take a look at these different divisions, and this will help us understand kind of a lot about them. You can actually see on this particular diagram that not only do we list them as parasympathetic and sympathetic, but we start to give them other names. We have craniosacral as an anatomical name for the parasympathetic division, and thoracolumbar as an anatomical name for the sympathetic division. And this is going to relate to where the actual nerve transmission comes from. Craniosacral means we have cranial nerves involved. The cranial nerve number 3, the oculomotor nerve, the facial, glossopharyngeal, and vagus nerves as well, and also a small component of the trigeminal nerve. Then we have S2 to S4, that's the sacral component. So there are portions of the parasympathetic division that come from the spinal cord. The sympathetic division is thoracolumbar. So from this T1 to L2 region of the spinal cord is where all of the sympathetic division impulses are going to originate. So let's go ahead and take a look at the various names, starting here with the sympathetic division. The sympathetic division has a functional or layperson's name of fight or flight. Some people like to add an additional F to this, freeze, just to indicate that when we think about fight or flight responses, this is going to be something that is going to be largely stimulatory, although neither one of these divisions is entirely stimulatory or inhibitory. But it's going to be largely stimulatory, doing things like raising our heart rate for fighting or fleeing. Sometimes this does just cause us to freeze in place. The anatomically based name of this is thoracolumbar, indicating where we actually have the cell bodies located, and they are in the lateral horns of the spinal cord from T1 to L2. Again, that's where we derive the term thoracolumbar. So if we think about the thoracolumbar division and we want to think about how it gets to various places, we do arise from T1 down to L2. We have ganglia on either side of this vertebral column. These are called paravertebral or sympathetic trunk ganglia. We can travel up and down these or directly out from them to any target organs that happen to be in that region. To get to something in the head like the eye, we have to travel up through other ganglia within the cervical region. And of course, we have some ganglia that are lower down, allowing us to access nerves that go out through the sacral foramina. But that's the basic component of it. You'll see that there is an additional set of ganglia here. These are not located off to the side. They're actually located anterior to the vertebral column, so anterior to the vertebral bodies, and they're going to be below the diaphragm, with the celiac ganglion being relatively close to the diaphragm, the superior mesenteric ganglion being relatively close to the superior mesenteric artery, and the inferior mesenteric ganglion being relatively close to where the inferior mesenteric artery comes off of the abdominal aorta. So the two types of ganglia we have then are sympathetic trunk or sympathetic chain ganglia. They can also be called paravertebral, meaning they are on either side and paralleling the vertebral column. And so we just recognize those as being parallel to the vertebral column. They are, of course, immediately connected to those above and below, so we're going to include those within that. The other three that we talked about are called the collateral, or I actually prefer the term prevertebral, meaning they're in front of the vertebrae. And these are anterior to the abdominal aorta, which runs along the anterior side of the vertebrae below the diaphragm. So we want to think about how we get kind of from one location to another, and this is going to relate to our spinal nerves. Of course, we know that we are going to initiate these commands if we're coming from the spinal cord rather than, say, from the cranial nerves in the case of the parasympathetic division. Talking about the sympathetic division itself here, we come from the T1 to L2 sections of the lateral horns of the spinal cord. We exit on the anterior root, get onto the spinal nerve, and we take this white ramus communicans here down to a sympathetic trunk ganglion here. And then we can synapse in that, or we can travel through that without synapsing and synapse elsewhere. You'll notice that this white ramus communicans, which is going to carry signals from the spinal nerve to the sympathetic trunk, which consists of the ganglia and the fibers that go up and down in between adjacent ganglia, that white ramus communicans is thicker than this gray ramus communicans. The gray ramus communicans is a way to get from the sympathetic trunk ganglion back onto the spinal nerve to head out to wherever that spinal nerve goes. That means if I want to innervate a sweat gland, I'm going to need to get to the skin. I need to take a spinal nerve to get there. If I want to get out anywhere to a limb, I'm going to have to actually get onto the brachial plexus, or I'm going to have to get onto one of the nerves from the lumbar or sacral plexus to be able to get down to one of those nerves that goes into my arms or my leg. I have to get onto a spinal nerve to do that. So we do know that this gray ramus will allow things to get back onto the spinal nerve if they're heading out to those locations. So let's think about impulses destined for areas outside of T1 to L2 actually get there. We want to think about kind of a pathway here, and we're going to basically describe that pathway. We, of course, originate somewhere between T1 and L2. All of the sympathetic division signals originate in lateral horns from T1 to L2. We exit the spinal nerve on the white ramus. So if we think about how this is going to work, we leave the gray matter from the lateral horn, we get onto the anterior root. In the intervertebral foramen, that joins with the posterior root to become the spinal nerve. We then, very quickly after that, exit on the white ramus communicans, and that's going to allow us to enter the sympathetic trunk and to get to a sympathetic trunk ganglion. In this case, let's say we want to go up to the head or somewhere like that. We need to get out of the T1 to L2 region, so we travel upwards from that ganglion on the sympathetic trunk. Now, every spinal nerve has a gray ramus, so if we get to the cervical region, and let's say I need to go out and send a signal to the axilla so that I can stimulate sweat glands in that region, I can go ahead and hop onto one of those cervical spinal nerves that's part of the brachial plexus and carry that signal out that direction to be able to initiate sweating from those sweat glands. Now, of course, if I'm going to go up to the eye, I'm actually going to go out through the top of those cervical ganglia and head directly to the eye rather than hopping on a gray ramus and then traveling out a spinal nerve. But those are the possibilities that we have. So let's take a look at some of these pathways and imagine how we would get to various locations. You can see here for this first spinal nerve pathway where we exit that lateral horn, go out that anterior root, get on the spinal nerve, hop off on the white ramus communicans. We can synapse or not synapse within this ganglion. If we do synapse there, we have the potential to hop out and head out through this gray ramus communicans to get onto that spinal nerve, going out to something like an erector pili muscle or out to a blood vessel. And so we can hit those targets by using that gray ramus communicans. If we're going to go to an organ like the heart, where the sympathetic division might want to speed up the rate of contraction of the heart, we're going to have the same initial component of this. We're going to start in a lateral horn, go out an anterior root, off on a white ramus communicans, entering a sympathetic trunk ganglion where we either synapse and go directly to our target organ, not on a spinal nerve, or we can actually travel to a different level of the sympathetic trunk and synapse there and have a nerve that goes out to our target organ. So in either case, we're not using spinal nerves. Now we are in roughly the same area as our target organ, and we can leave the sympathetic trunk ganglion and go directly there. Now it ends up that there is another pathway, and it's going to involve some other ganglia. So we have what we refer to as splanchnic nerves. Splanchnic is another term for visceral, meaning organ, and these are nerves that are going to carry axons which have passed through a sympathetic trunk ganglion without synapsing to allow them to get to the collateral ganglia, where they will synapse prior to sending their postganglionic axon on that ganglionic neuron to the abdominopelvic viscera, the organs in the abdominopelvic region. And if we take a look at how this works, we're going to again start in a lateral horn from T1 to L2, exit out the anterior root. We're going to get on our spinal nerve, off on a white ramus communicans. We either pass directly through a sympathetic trunk ganglion and out onto a splanchnic nerve, or we first go to a different sympathetic trunk ganglion and pass out on a splanchnic nerve. And then we get to our prevertebral ganglion, and then we're going to have our ganglionic neurons extend out from there to our involuntary effectors, in this case, the musculature of the intestine. So what we can see is the splanchnic nerves are those nerves that are filling that space in between the sympathetic trunk ganglion and the prevertebral ganglion. All right, so let's kind of learn the names of these three collateral or prevertebral ganglia. They are all named for arteries which are in those same general locations, so learning these names will actually be helpful now. We have the highest up, the celiac ganglion, which is near the celiac trunk. The celiac trunk is an interesting artery we will learn that gives rise to the common hepatic artery, the left gastric artery, and the splenic artery. Then we have the superior mesenteric ganglion. You can see that this ends up sending some nerves to areas like part of the large intestine and the small intestine, and that would be the same general area that the superior mesenteric artery provides blood to. We can see the inferior mesenteric ganglion here is going to innervate things down here like the bladder, like the rectum, so it's kind of towards that end of our large intestine, and this is some of the same area that the inferior mesenteric artery provides blood to. Therefore, these names do matter for us, and we will come back to them later on in the semester. Now, as far as our neurotransmitters, again, we recognized earlier that the sympathetic and parasympathetic divisions share the use of acetylcholine, and that acetylcholine is going to happen here in the ganglion. But when we get out to our effector, the sympathetic division is going to use norepinephrine, whereas the parasympathetic is again going to use acetylcholine. So kind of important to kind of recognize what's going to happen here. Norepinephrine is also called noradrenaline. It's kind of another chemical form of adrenaline, which we simply call epinephrine. And so these are two chemicals which tend to largely be stimulatory, at least in regards to things like heart rate. So this is kind of what we're going to have going on here, and we're going to contrast that again with the parasympathetic division. Now, we do have some really important component here associated with adrenaline or epinephrine, and we have this link between our neurotransmitter at most of our effectors, the epinephrine, and the ability of the fight or flight response to result in a hormonal response producing a release of adrenaline. This comes from the adrenal medulla. And of course, here's our adrenal gland sitting on top of the kidney. The adrenal medulla, with medulla meaning middle, would be the middle layer of this. And it's going to work differently, of course, than the adrenal cortexes, which is what we see from the outside. So this is an endocrine gland. It is stimulated through the adrenal medulla pathway. Beginning again in lateral horns, we come out on our anterior root. We head to a white ramus communicans off that spinal nerve, go through this sympathetic trunk ganglion, take a splanchnic nerve, do not synapse in a prevertebral ganglion, head directly to the adrenal medulla, and then we're going to have some modified neurons producing the epinephrine and norepinephrine within the adrenal medulla. It is mostly epinephrine, but norepinephrine is also produced. Now, because this is an endocrine gland, we are going to release these into the bloodstream. And this kind of brings up an important point about the difference between, say, the autonomic nervous system and our endocrine system. The autonomic nervous system in this case is going to lead to the production of adrenaline. The autonomic nervous system, in the case of the sympathetic division, is going to do things like stimulate our heart rate, prepare our muscles and brain for higher levels of activity, and this adrenaline release is going to do functionally the same thing, but because it's going to circulate throughout our bloodstream, it's not going to turn off as quickly. So it'll leave us with kind of a jittery, elevated state after maybe we've had some kind of fear response because of all of this adrenaline floating around through our bloodstream. So we want to think about these effects of the sympathetic division. We do get things like increased alertness through what we call the reticular activating system of our brain. This can produce some of the euphoria associated with things like thrill rides. It does increase cardiac and respiratory activity to be able to mobilize more oxygen. It increases our muscle tone. Again, if we're thinking about fighting or fleeing, we're going to need that. We get some energy mobilization so that along with the oxygen, we can fuel our muscles and brain. We dilate our pupils, allowing more light in. We constrict peripheral and visceral vessels. This is kind of an interesting one because we could kind of consider to be inhibitory in that we are not providing blood to these regions. Obviously, we have to stimulate some of the smooth muscle to be able to actually contract those and provide less blood to these regions. But if we're talking about less blood to our skin and less blood to some of our organs, particularly digestive organs, this makes more blood available to our brain and muscles. So it is part of this overall stimulation for this short-term response to maybe fear. We then stimulate sweat and erector pili muscles. And this leads to ejaculation in the shoot part of what we refer to as a point and shoot system. So you might think about sexual response as being relatively complicated. Point and shoot makes it relatively easy. Shoot for sympathetic, point for parasympathetic. Pointing means the arousal that leads to erection. And then, of course, the shooting then is the ejaculation. So we can associate these with the letters that are in our sympathetic and parasympathetic divisions to remember this kind of mixed response for sexual activity. And that's going to take us into the parasympathetic division, which again, we are trying to contrast with the sympathetic division. The functional or layperson's term for this is rest and digest. That's pretty much the opposite of the sympathetic division. The sympathetic division is called fight or flight, but if we think about what it did, the sympathetic division also shut off some of the blood flow to our organs. Obviously, the parasympathetic division is going to increase that blood flow so that we can digest. Anatomically, this is referred to as the craniosacral division, indicating that we have cranial nerves involved, anything that has an involuntary component to it. And then we have the sacral component, the S2 to S4, that we see down at the bottom of the spinal cord. Now, of course, I know you're thinking, well, there's really no spinal cord in S2 to S4, but we're thinking about those roots from the cauda equina, which exit from that location. All right, so where are the cell bodies located? Well, they're in the brainstem if they're going to be associated with cranial nerves. Again, it's cranial nerves 3, 7, 9, and 10. And they're going to be in that S2 to S4 section of the spinal cord, or where the S2 to S4 nerves arise, if we want to think about it in that manner. So what cranial nerves transmit these impulses? We're going to kind of skip the fact that there is a small component of the trigeminal nerve doing this. We want to remember the trigeminal nerve largely as a nerve which carries sensory information from the face back to the brain. So we'll focus on things like the oculomotor nerve, the facial, the glossopharyngeal, and the vagus. And we can think pretty easily about what each of these does. So if we think about the oculomotor, we're going to the eye and we're doing something involuntary to this. We know that the sympathetic division was going to dilate the pupil. This will do the opposite. It's going to constrict it. It's also going to control the muscle through this ciliary ganglion we see labeled up here, called the ciliary muscle, which adjusts the lens of the eye. The facial nerve we already kind of know, as we do for the glossopharyngeal. Both of those are associated with taste. And when we think taste, we also think about our saliva production, which of course is going to be an autonomic thing. It's involuntary. So the facial nerve is going to innervate the submandibular and sublingual salivary glands, where the glossopharyngeal innervates the parotid salivary gland. The facial nerve also innervates the lacrimal gland, the gland associated with your eye that produces tears. The interesting and really big crazy one here is the vagus nerve, which we see right here, coming off, going to the heart, going to our breathing tubes, going to organs down in the abdominopelvic region. Now, of course, the pelvic component primarily coming from the S2 to S4 through this, what we call hypogastric plexus, working its way to things like the rectum and the other parts at the end of the colon, the descending colon and the sigmoid colon, which we can't really see here. So the vagus nerve hits pretty much the rest of that. It's a very, very large nerve. It's a large cranial nerve, but it's also just kind of going to very, very large amounts of area within the body. And again, as a cranial nerve, the vagus nerve was very different in its target, meaning its targets were primarily outside of the head. So if we think about another division or difference between the parasympathetic division and the sympathetic division, we have that difference in neurotransmitters. We use acetylcholine completely here, both in the ganglion and at our target. Now, of course, this can stimulate or inhibit depending on exactly what we got going on. And in physiology class, you'll learn more about the different kinds of receptors that might be on the other end. There are ganglia that are associated with the parasympathetic division. If we think back to the sympathetic division, it was the paravertebral or sympathetic trunk ganglia, as well as those prevertebral or collateral ganglia. Now we have what we call terminal or intramural ganglia. What's more important here is that in the parasympathetic division, the ganglia are either next to, that is right near, or in the wall of the actual target organ. So the sympathetic division had the ganglia relatively close to the spinal cord. If we think about how this works, we can kind of get an idea of a sensory response leading to an autonomic result in the parasympathetic division. We have this urinary bladder which is collecting urine and starts to stretch. This is going to stimulate some of the visceral receptors there, which send a signal back going through this posterior route to that posterior horn, and the spinal cord will receive this information, and an interneuron will carry this to that lateral horn, and we're going to have our autonomic impulse head out from there, and it's going to work its way out all the way on its preganglionic axon until it gets within the wall of the bladder. That will deliver the information to the postganglionic axon, and that's going to stimulate the smooth muscle here of the bladder to contract to cause us to either urinate or to need to urinate, and we can use this skeletal muscle sphincter down here, the external urinary sphincter, to avoid urinating if we wish to. So let's think about some of these effects. We expect the effects of the parasympathetic division to essentially be the opposite of the effects that we saw for the sympathetic division. We're going to constrict pupils. We're going to secrete from our digestive glands and increase gastrointestinal motility or movement within our gastrointestinal tract. All of this is stimulating digestive functions, the exact things we would shut down if we were having a fight or flight response. We're going to constrict respiratory passages, and we're going to reduce heart rate. It's the opposite of what we had the sympathetic doing. The sympathetic was going to increase heart rate, and it was going to dilate respiratory passages to allow more air in. We, of course, can't keep our respiratory passages open and relatively mucus-free all the time, or else we're going to inhale a lot of particles down into the delicate parts of our lungs. And as far as the sexual response, this is responsible for arousal or erection, increasing blood flow to the genitals. So it's the point part, if we think about erection, of that point and shoot system. Again, point standing for parasympathetic and shoot standing for sympathetic. So we want to think a little bit about some more complicated responses and how we regulate homeostasis, or that constant internal environment in our body. We know we're going to use the autonomic nervous system for this. So we do have a region of our brain which is a control center for homeostasis. It works using the autonomic and endocrine systems to help maintain this. And it also is sometimes responsive to emotional states, meaning that we don't always maintain homeostasis, but instead we may respond to emotional states using this region of the brain. This, of course, is the hypothalamus, which we already knew was the control center for the autonomic nervous system and endocrine systems. This is responsible for things like pupillary reflexes in regard to light change. We can, of course, have fear, an emotional state, result in part of our limbic system, a particular part referred to as the amygdala, stimulating the hypothalamus, and then the hypothalamus initiating a sympathetic or fight or flight response, even though there's not actually something to respond to in all cases. So we can have fear that isn't really associated with the danger produce the same kind of response of fear that would be associated with danger. But it again is linking the limbic system to the output of the autonomic nervous system. Now, we do also have some control within the medulla oblongata. In particular, we think about the cardiac control. So the region of the brain that has nuclei called the cardiovascular centers, which is going to control smooth and cardiac muscle, smooth muscle of course, vessels, and the cardiac muscle of the heart. That is the medulla oblongata, and this helps us to regulate heart rate and blood pressure. Now, if we kind of think about what's going on here, you can see that we've got the vagus nerve being able to carry information to the heart in the parasympathetic system. Parasympathetic will decrease heart rate. But we can also have, coming from this area, sympathetic cardiac nerves, which work their way down to the heart, and of course, these are going to increase heart rate. As far as control of the autonomic nervous system, we recognize control through the parasympathetic and sympathetic divisions, but there's really one more. We have kind of an independent aspect of our nervous system, sometimes thought about as separate from the ANS, sometimes thought about as part of the ANS. This is the enteric nervous system, which is active in the digestive system. So in the case of the digestive system, we have kind of this communication between different parts of the digestive system, which is working independently of the brain, working independently of the spinal cord. And for example, we can think about the idea that if our stomach stretches, we're going to need to have communication of that sensory signal with the glandular production of the stomach so that we can actually produce some of our digestive gland or secretions. And this is a local communication. Again, it does not involve communicating back through the central nervous system. It's not a reflexive response in that way. It's all a local set of information completely within the digestive tract and autonomous, that is, working within itself as opposed to being controlled by anything else.